So, hello everybody again. It's great to see you all and so many of you joining us for today's talks about dragonflies and damselflies. My name is Anja and I'll be hosting this event on behalf of the association Hyla. And for those of you who are not familiar with our work, I'll give just a brief introduction before Maja's lecture. So about us, we are a non-governmental organization. We were founded 29 years ago by biology students, and they noticed at that time the lack of data on creations of amphibians and reptiles. And they wanted to do something about that and also actively support the conservation of those species and their habitats. From those enthusiastic and humble beginnings, we are now a diverse team of biology experts, and we have many different research and conservation projects all over Croatia, also in some neighboring countries. Over the years, we also expanded our focus to include not only amphibians and lizards, but also some insect groups we became interested in. This means that in the fieldwork season, you can find us basically all over Croatia. We'll be studying toads and newts in various wetlands. We'll be climbing rocky terrain, searching for snakes and lizards, casting research nets for freshwater turtles, waving entomological nets, inspecting vegetation for butterflies and their caterpillars, setting traps for beetles, even kayaking along rivers in search of dragonflies, all with the goal of having a solid scientific foundation with sufficient data, so the conservation measures we create and propose for implementation to the relevant Croatian authorities are aimed at the best possible protection of the species and their habitats. Part of our mission is not only to constantly build on our knowledge, we also like to share that knowledge and discuss it with the scientific community and the public, and we encourage that they become actively involved. One of the questions we got in the registration form for this lecture was, how can you contribute if you become our member? Membership in Hyla depends on your individual interests and availability. Some members just pay the membership fee each year to show their support without much involvement. Some members choose also to volunteer by joining us in the field, or they help with educational activities. Some write articles for our blog. Some work as external experts on some of our projects. Citizen science is also a way you can contribute. For that, we recommend Biologger app. We use it in our work, but it's free. It's open to all. It's similar to eNaturalist. It allows you to record the species you encounter. But unlike eNaturalist that has this community confirmation method, Biologger has admins. These admins are experts for our local fauna, and that's making the data more reliable for use in further research or in educational materials. Some of Hyla's experts are here with us today, and they will also be available if you have any further questions about our work. And if you don't already, I hope you will drop us a follow to stay in touch, and maybe our posts will spark an idea for some future collaborations. You never know. For example, today's lecture is a result of us connecting with Maja. She was following us on socials. She saw one of our posts about river clubtail dragonfly, Stylurus flavipes, and she asked if she could have its exuvia. We were curious about this enthusiastic person and, of course, happy to share this exuvia. Maja came to our office to collect it, and we got to talking. She is so easy to talk to. We found common interests. Maja became our member. Then we met on another occasion when Maja was visiting Zagreb. We went for a nice walk to Lučko lakes. And eventually we got this idea for a lecture. Her passion for talking about biodiversity, learning about dragonflies, sharing the knowledge is inspiring. And we wanted to share this amazing experience of Maja with all of our members and followers. And we are so grateful for her time and for her willingness to do this lecture today. As you can see, Maja has done some impressive research, and she's worked at some of the Europe's top institutes. Her research mainly focuses on water fleas, Daphnia, but she was also a part of projects such as Grassland Resilience as data analyst and R programmer that was at Queen's University in Belfast, as well as food webs across aquatic terrestrial environments project that was at Swiss Federal Research Institute, WLS. After that, she did eco-evolutionary dynamics research at University of Innsbruck. And today, she holds the position of research associate and is also teaching at the University of Cologne. On top of it all, Maja started a passion project focused on dragonflies and damselflies in urban garden ponds, which she will talk about today. You can also follow it on Instagram. Here's the QR code, so it's easier to track. And without further ado, Maja, I will let you take over. I would just like to ask all of you to please keep your mics on mute and submit any questions you may have through chat. We'll try to answer them all after the lecture. If it's a quick question, we'll answer it immediately in the chat. Thank you all for coming and enjoy.
Thank you so much, Anja, for the very kind introduction. It is really a pleasure to be here, and thank you also to Hyla for giving me this opportunity to share my passion for dragonflies with all of you. I hope you can all see my slides. I will just give it a go. Please shout if anything is not working properly. I hope that's not going to be the case. But yeah, we'll just see how it goes. So today I would like to focus mostly on Odonata in urban garden ponds, which was a project that I did last year. But I would first want to give you a short introduction about dragonflies and damselflies, tell you a little bit about their life cycle. Then I will move on to the living garden ponds in cities, and then we can finalize this meeting with a Q&A session. I will also share a few QR codes along the way, so feel free to take a photo. And if you have any questions, as Anja already said, please just post them in the chat. So let's first start with the introduction to the Odonata. They belong to insects. It's an order of Odonata, which we can split into Anisoptera. English, that would be the dragonflies, and in Croatian, Develostruka vretenaca. In Germany and in Croatia, which are the two countries where I kind of have my interest in and focus my research on, we can find 5 different families of Anisoptera, of the dragonflies. And then we have the Zygoptera, which are the damselflies, or Tankostruka vretenaca. And here we have 4 different families that occur in Germany and Croatia and in most of Europe. And before we continue, I would like to talk about 2 myths. Basically, it's questions that I get asked quite often. And one of them is if dragonflies can sting. And I can tell you, no, they can't. So you shouldn't be afraid of them. And the second one is they can bite. And again, it's kind of a yes and no answer because it feels more like a pinch if they do so, but it really doesn't hurt and it's not dangerous. So of course, if you annoy them, which you should not do, they might bite you quickly, but it's not going to be anything that's going to be painful. So I think the conclusion from this is that dragonflies are actually harmless, so you don't have to be afraid of them. Now back to the dragonflies, the anisopterans. What kind of separates them from the damselflies is mostly their stout and robust body. So you can tell they're usually much bigger and very robust. Their hind wings are broader at the base than the forewings, and they usually spread out at the right angle when they are just resting, for example, on a plant stem. And their eyes are quite large and mostly not fully separated. So they either meet across an entire line or just at a single point. The only exception here are the Gomphidae, where the eyes are actually also separated, but at least not as much as they are in the damselflies. And one really cool fact is that these compound eyes that the dragonflies have can have up to 30,000 individual eyes, which is really absolutely insane. On the opposite, we have the Zygoptera, the damselflies. They usually have a rather slender and delicate body. Their eyes are clearly separated, so they're really on the sides of the head. And their forewings and hindwings are of equal size and form, and they're usually folded directly along the body. Here, the exception would be the family of Flestidae, where the wings are also spread in kind of a right angle when they're resting. One thing that is quite important to know and understand about Odonata is their role in the aquatic terrestrial food webs. And as some of the other insects that have larval stages, dragonflies undergo this life cycle that has this aquatic and terrestrial part. So they live in the water as larvae, and at some point they emerge, and then they live mostly in the terrestrial part, although they come back to the water for reproduction, for example. As larvae, they live as predators in the water, and they feed on other invertebrates, but also on other dragonfly larvae, so they're also cannibalistic. They do also feed on fish larvae and, for example, tadpoles. At the same time, they also serve as food for larger fish and, for example, amphibians. Once they emerge as adults, or the scientific term would be imagos, they're very skilled hunters and feed on various insects, including mosquitoes, flies, and butterflies, but they're also an important food source for spiders, bats, birds, and similar. One thing that is also not a very common knowledge, I would say, is that such insects like dragonflies with these larval stages that develop in water, in the aquatic systems, that they're also of a high nutritional quality for their predators because they're usually rich in essential omega-3 fatty acids, which for many animals are important for their growth and reproduction, but most of animals cannot produce these fatty acids de novo. So this is where the role of especially the imagos, the adult dragonflies, becomes even more prominent for the rest of the food web. Now, coming to the actual life cycle, I'm going to start here with the oviposition. Again, scientific term for egg laying. So depending on the species, these egg laying can take place either without or with the accompaniment of the male, which is in the so-called tandem position. And it happens either directly in the water, so exophytic outside of a plant, or in the plant material, which would be entophytic inside of a plant. This is then followed by the larval stage, which, as I said already, is completely taking part in the water. The first stage after the hatching from the egg is called the prolarva. After that, the larva molts several more times until the emergence. Depending on the species, this can take something between 3 months or so, or even less, but also up to 5 to 7 years. So a larvae can actually stay in the water for a quite long time. After the larval stage, what follows is the emergence. Dragonflies and damselflies both are hemimetabolous, which means that they develop directly from the larva to an imago, to an adult dragonfly and damselfly without the pupal stage. So as an example, the opposite would be the holometabolic development, where the larvae first develops into a pupae and then into an adult organism. An example for that would be the butterflies. So the emergence in dragonflies works in a way that they climb out of the water, mostly along a plant stem. The only exception here, again, would be the Gomphidae, and then they molt for one final time. And what remains from this molt is the so-called exuvia. You can imagine this as some kind of a shedded skin, which is just the exoskeleton of insects. In this case, refers also to dragonflies and damselflies. After the emergence, the freshly molted imagos first pump the hemolymph into their wings and the abdomen. And this causes the abdomen and the wings to extend. And at first, the imagos then also appear quite pale, so they don't have the proper coloration, which then develops over time. It is really important that if you find any dragonflies or damselflies in the nature where you can tell that they have this pale color, and you can also see that the wings kind of have a weird reflection, you can tell that these are very freshly emerged individuals, and these are very fragile at that moment. So they should be left alone. You should not touch them or come near to them and not disturb them in any way, because they're quite vulnerable in this stage. What follows after the emergence is then the mating. During the mating, dragonflies form a mating wheel, which is very, very specific for this order. They do it with their bodies, and it actually almost looks like a heart, which is quite cute to see. In this process, the male grabs the female either at the back of the head or at the pronotum, and then the sperm is transferred via the male's secondary genitalia to the female. And after that, then follows again the egg laying, which I mentioned at the very beginning. I'd like to spend some time showing you some of these very important kind of morphological characteristics of dragonflies and damselflies, because I find them extremely fascinating. So I already mentioned the mating part. And what is important to know here is that the males actually have two sets of genitalia. So they have the primary genitalia here on the left, where the sperm is actually produced and then transferred to the secondary genitalia. And that is based usually at the very base of their abdomen. While in females, at almost the same position on their abdomen, they have the ovipositor, which is basically the egg-laying apparatus. Compared to the females, which don't have anything else, they only have this part that is for the egg laying. The males have also the secondary genitalia, which I mentioned earlier, and these are then higher up on the body, on the second or third abdomen, and are used for the actual process of mating and also for the sperm removal. So it also happens that males that are mating with females that have already mated with another male can actually use their secondary genitalia to remove the sperm of the male that was there before them, which is, again, quite fascinating. And this is how the combination looks like. So what is shown here is basically the secondary genitalia of the male, which they can also extend slightly. And then the bottom part here is showing the ovipositor of the female. And there is actually even one very, very tiny egg here that belongs to the female as well. As mentioned earlier, the base of the abdomen of the male then usually grasps the female. In dragonflies, it happens at the back of the head of the female, and in damselflies, it happens on the pronotum. What I find super fascinating to see is really this stage when the dragonflies just emerge. And you can imagine just this, having this larvae, and then just a few moments later, seeing this adult dragonfly coming out of the exuvia, out of this larval skin. And I was really happy to see some of these emergence occasions in the wild. And I took a few pictures, and this is one that I really love showing because it shows how tiny and small these wings are before the dragonfly starts pumping the body fluid, the hemolymph, into the wings. So these are already kind of, the only structures that you can actually see. in the larvae, or like in this case, in the freshly emerged imago, which then extend to this proper, really nice wings just by pumping the body fluid into them. The exuvia, we mentioned them already earlier. What I really find absolutely fascinating about them is that A, you can easily collect them in the wild, which, however, as a small note, is not allowed in Germany, as far as I know, without permission. So be careful about that. But if you do collect any, you can actually use them to fully identify them down to the species level. This acquires a little bit of practice. There are quite some good guides and identification keys that you can use. I'm also currently working on an app that will allow you to do so on your phone or on your tablet. I will introduce it sometime later in the talk. But this is actually how my, let's say, passion for dragonflies and this like huge interest started. It was really just by collecting exuvia, taking pictures of them, trying to identify them all to species level. And it's something that is really a lot of fun. You learn a lot. You learn it very, very fast. And it's a really great non-invasive method for assessing the species richness and in general the diversity of the dragonflies and damselflies. And sometimes it goes down to really, really tiny characteristics that are very specific for certain species. In this case, for Enalagma certigerum, it's a really, really tiny kind of horn that is next to a small bristle on the movable tooth. So it's really, really crazy how specific it can get, but still it's really easy to learn. What is also possible is to actually separate the exuvia by the sexes. So it's very easy to see what are the female exuvia and the male exuvia. Here on the bottom, on the very left, we see an exuvia of a female Aeshna cyanea, where you can clearly see the ovipositor, the egg-laying apparatus, or like the remains of it. And in the case of a male exuvia, you can actually see the remains of the primary genitalia and the secondary genitalia. And as Anja mentioned earlier, it's actually thanks to the exuvia that we connected with each other. This was exactly the sample that I got from Hyla back then. And I was extremely happy to have a few of these and took photos of them. And I'm posting every now and then some of the photos with the most important characteristics. So again, if you're interested in seeing more of these exuvia and learning about them, then feel free to follow me. What I find, again, very important to mention is basically the habitat where we can find dragonflies. And we can find them even in garden ponds, as I will show later. Most important is we need water. We need water, we need vegetation, and we need to protect these habitats. And this is one of the reasons why I really love doing this, talking about this, making people aware that the species protection actually starts with habitat protection. And this is something that us scientists, us biologists, ecologists, call us however you want, we need to put a lot of effort into that as well. And we need to basically make it kind of aware to everyone else how important it is to just protect the environment around us, because that's the habitat that we live in, but also the habitat where all of our beautiful species call their home. There was one specific habitat that I once visited in Switzerland, which is here on the bottom left. I did it during the project that I did there with my PhD student, with Bastian. And it was actually a really small puddle that I suppose basically just happened to be there because there was probably someone with a tractor or something else that went through this meadow and just left tracks, and those tracks then filled with water. And even that small puddle then became a habitat for Odonata. So we were there and we actually found even a small snake. We found a few small frogs jumping around, but we also found dragonflies there. So this is something that I found super amazing to see, that even this small puddle, just small part of water, can actually become a habitat for Odonata. If we now compare the number of species that we can find in Croatia and Germany, we can see here that there is actually a bunch of species that occur in both countries. So according to what I found, it's 63 species that are shared between Germany and Croatia, and then there is 19 more species that we find in Germany but not Croatia, and 10 species that are found in Croatia but not in Germany. Germany is a bit more in the north. It's in general a bit colder, so we have a few species there that are probably more adapted to colder environments, while Croatia, as we all know, it's in general a rather warm country where we probably find a bit more of the species that are kind of warm adapted. But as we know, climate change, this all can lead to a very different distribution. So let's see what's going to happen in the next 5 to 10 years. These are the 10 species that we can find in Croatia, which do not occur in Germany. And my favorite out of those is the Lindenia tetraphylla. In English, it's the blade-tail. In German, it's Seedrache, which translates to sea dragon, I would say, which I absolutely love as a name. And in Croatian, it's Jezerski regoč. I've never seen it before. I'm really thankful to Hyla and to Borislaus for providing these pictures. I find it such a beautiful species, and I'm really, really hoping to see it one day. I would then quickly want to share some data from the red list of Odonata in Germany and Croatia. It's important to mention here that the categories don't really entirely match to each other, and that especially the German categories do not entirely match the official categories of the red list. But we can still clearly see that the majority of the species is of least concern, which is here the really huge kind of green bar. But we also do have several species that are either rare or already critically endangered or endangered. So there is definitely potential in protecting these species. And if you want to find out more, there are these 2 publications that I looked into, which actually summarize the status of the dragonflies and damselflies in Croatia and Germany. As I mentioned already earlier, in Germany, all of the species and their habitats are protected. So in this case, this refers to several other countries like Austria and Luxembourg and Spain. So there, if you want to do anything with dragonflies or damselflies, you need a permission. While in Croatia, for example, only the species of the Habitat Directive are protected. If you should have any questions to the Habitat Directive or similar, I hope that Hyla can answer those, because for me, this is also rather new territory, but it was just very important for me to mention this here. One of the species that is currently nearly threatened in Germany and actually vulnerable in Croatia, and therefore strictly protected, is the Seriagrion tenellum. And what I find fascinating about this species is actually that we find it here in Cologne. We have experimental gardens just behind our university with several ponds. And I've been there throughout this year, just like checking on the dragonflies and damselflies. And in several of these ponds, we could find larvae, but also freshly emerged adults. And I've also observed mating. So this was really, really great find, given that it's a nearly threatened species. And I can tell you the red point here is the location of our university. And you can already see that it's one of the rather southern kind of limits of the distribution. There is a few observations that have been done in the very south of Germany. But otherwise, if you just look at this area where we find most of the species, then this find is kind of at the more southern limit. Okay, so much about the introduction, and now I'm going to move forward with my project on the living garden ponds in cities. So why I did this project actually is because I took a course this year, which was organized by CanArt. CanArt is a nationwide initiative for training in species identification. And it's a project by NABU Nature Conservation Station Münsterland and the University of Münster. It is funded by the Federal Agency for Nature Conservation, with funds from the Federal Ministry for the Environment, Nature Conservation, Nuclear Safety and Consumer Protection in Germany. Its duration is actually for 6 years. So it started back in 2020 in April, and it's running only until March this year. And they had a really great funding of 1,800,000 euros. And what they offer are basic, advanced, and in-depth courses, which you can translate to something like bronze, silver, and gold courses on grasses, mosses, hymenoptera and wild bees, beetles, and dragonflies. What I find extremely amazing about this project is that they offer really free courses, except for, if there are excursions, then you pay yourself for them. But the courses themselves and all the material that was shown to us and everything else was free. And they organize them a few times in the year at different places in Germany. And they basically recognize that people who are really good taxonomists and people who can identify species well are those that we need in nature protection, but we do not have many of these people. Like, these people are kind of, I don't know, the numbers are kind of decreasing. We don't teach this properly at universities. So we're really missing this part quite a lot. And this is why this initiative was actually called into place to offer people who are biologists, but also people who are maybe just like very interested in nature protection. You don't have to have studied biology for this. They offer these courses, and I'm really, really happy that I was able last year to actually take part in one of the in-depth courses for the dragonflies. And within that course, we all had to do a small project. And this is how my urban ponds project happened. Okay, so about the research questions and aims of the project. I first wanted to look if the diversity of the larvae that I find at the points matches the diversity of imagos. Reason for that is that we know that obviously the adult dragonflies and damselflies can fly around, while the larvae, they stay at the same pond. So I really wanted to see if there are any species that are kind of, visitors that are maybe coming from somewhere else, or if there's a lot of species just leaving those ponds and that they only stay in the ponds as larvae. So that's why this comparison was kind of one of the main questions of this project. The second one was to look into the species-specific phenology. Phenology is basically kind of the process of the life cycle throughout the year, let's say. So when do they develop as a larvae? When do they emerge? When do they mate? When's their flight season? All of that basically belongs to the phenology. And I just wanted to see if we have any differences between the species. Then I was interested in seeing which of the species actually coexist in the ponds. And finally, which of the abiotic and biotic factors and also pond characteristics are actually important for the dragonflies and damselflies. I did this project in an allotment and leisure garden association in Cologne, which is a few kilometers away from the center. It's called Kleingartenverein Flora. It's in the part of Cologne called Nippes. And here I selected 10 ponds that were different in size. Out of those 10 ponds, 5 were linnet ponds, 3 tub ponds, 1 barrel pond, and 1 metal tub. There was only 1 pond with fish, but in several ponds I found frogs and newts, for example. I was able to visit these ponds between 2 and 4 times between May and August last year. I did plan a few more visits, but that was not possible due to different reasons, being vacation or bad weather. So at the end, it was 2 to 4 visits per pond. These are just some of the pictures from the pond. You can see that we have a variety of sizes and also kind of the structures and vegetation. The pond, which is here on the top in the middle, is the one that had fish. All of the other ones were fish free. And it's this bottom pond here. Just pay attention when I change the slides now. The bottom pond in the middle. this is how it looked like in August. I'm going to go quickly back. So this was April, May, and this was then in August. So it was completely covered in, now I missed the word for it, but I'll come up later with it. I know the German word, I don't know the English one. So this was the biggest kind of change in, let's say, in the cover over the few months. But apart from that, of course, the vegetation changed a little bit, but most of the species were present at the time of the entire project. And these were then the remaining 4 ponds. As I mentioned earlier, we had this barrel pond, which is on the left, and the metal tub here on the bottom right. And on the top right was one of the smaller ponds as well. I'm really grateful to my colleague Johannes, who actually also took some drone photos of these ponds. And I'm only going to show 2 just to kind of give you an idea what kind of surroundings these ponds had. So there was one of the ponds which was here, and spoiler alert, it was one of the ponds with the highest diversity of dragonflies, which was really great to see. And then here we have another pond as well. This was actually the pond that had the fish inside. And this is the idea also for the future, that we kind of try to take more of these drone photos and also look into how far the different ponds are away from other neighboring ponds, which wasn't possible in this project, but it's something I would like to do in the future. So what I actually did in the project was to sample the larvae. So they were collected with a sieve. I was going through the pond with the sieve either 5 or 10 times, depending on the size of the pond. And then I photographed each larvae, but I did not take them with me to the lab. So the larvae were always basically just put back into the pond. So they were just collected and photographed. Exuvia were collected and later identified for a maximum of 15 minutes, again, depending on the size. Imagos were observed and recorded for 15 minutes, and for this it was really important to have good weather. Then I also measured some water quality parameters, such as the temperature, conductivity, pH, and O2, oxygen concentration and saturation. And then finally, I recorded the vegetation directly or later based on the photos. The larvae we identified based on the photos were possible. I can tell you this was very, very tricky for the larvae of the damselflies because they're really very tiny. And then I also identified the exuvia, and here it was possible to identify them down to the species level, including also the sex, either female or male. This is me during the project. Many thanks to those who were taking pictures of me as well. It's always nice to have this to actually show what I've done. This is also some part of the work that I did. These were, for example, the exuvia, which I then collected and brought them back to the lab, and then used a stereo microscope to take really good pictures and identify them to the species level. Okay, now coming to the results. When it comes to the species that I observed, I'm now separating them here into 3 categories, basically larvae, exuvia, and the adults, the imagos. I observed 4 different species of dragonflies: the southern hawker, emperor dragonfly, four-spotted chaser, and the common darter. In this case, when it comes to Aeshna cyanea, so the southern hawker, and Sympetrum striolatum, the common darter, I found all 3 stages, so basically larvae, exuvia, and adults, while for the 2 other species, I found either the larvae or the imagos. And then for the damselflies, I was able to observe 5 different species, again in kind of the different life stages. I observed all of them as imagos. The Pyrosoma nymphola, the large red damselfly, was the only species where I could find all 3 stages. For these Quenacronidae, so for azure damselfly, common bluelet, and the blue-tailed damselfly, I cannot really tell the difference with the larvae. So I cannot tell which exactly I found, but there was definitely quite a number of these that belong to the Quenacronidae family. So that's why I also kind of summarized them as such later on in the plots that I will show. These are just some pictures. So on the top, we have the southern hawker, modri kralj, and then on the bottom, we have the common darter or the veliki streljac. Always on the left is the males, and then on the right, we have the females. This was the large red damselfly on the top and the azure damselfly on the bottom. And I would say my favorite was definitely the common winter damselfly, zimska djeva. And the cool thing about this species is that it was totally unexpected to find it. And also that it's a species that overwinters. And as far as I know, in Germany, it's the only species that overwinters as an imago, so as an adult, while usually other species only overwinter as larvae. So this was really a great find, and I was quite happy to see it, but it was also only one single individual that I found. So now coming to the data, each of these panels is showing one of the ponds. The only pond where I didn't find anything was the metal tub. So the metal tub is excluded here from the plots, but all the other ponds are shown here. The different bars are referring to the different months when I visited the ponds, and they're basically just showing the number of larvae, in this case, that I found belonging to the different species. And you can already tell that there were quite some differences between the ponds. So, for example, the pond number 5 had a really large number of the Coenagonidae larvae, and only a few Pyrosoma nymphoa larvae, and I think just maybe one single larvae of Aeshna cyanea. So it was mostly the damselflies that I found. While I also had, for example, the pond number 7, which was quite diverse with a lot of larvae. However, you can already see here that there was not only a difference between the different ponds in which larvae we found, but also the difference in the kind of the composition of the larvae that slightly changed over the season, but not as much as, for example, the adults, which I will show in a bit. What I did with the larvae, which I didn't mention earlier, was that I also calculated their length, or basically measured the length of the larvae to see if over the season the average size of the larvae actually changes. And now here I'm showing the 4 species where I did so, or basically Coenagonidae as the family. And if you now focus only on the Aeshna cyanea, which is in the upper left part, you can tell that it seems like over time the kind of the average length decrease. So the average is basically the larger circle. But when we then look into the different ponds, and if we specifically look into these 2 ponds, number 7 and number 9, we can actually see that we had a certain switch at some point. So at the very beginning in May, there were quite a lot of really large larvae. So here the average was rather high. But then from June on, we started having a lot more of the smaller larvae that obviously then also grew over time. If you look into any of these other ponds, for example, in pond 2, in June, we still had some of the larger larvae. So here they just like kind of took longer to emerge. But in the pond 7 and 9, we can clearly see that between May and June was the time when probably most of these larvae actually emerged. And then new larvae develop from the eggs, and that's why kind of we have this switch in kind of the length and the body size structure between the basically within the project. Now coming to the exuvia, again, we can see differences between the ponds. There are a few ponds that only had up to 2 or 3 species that I could find, or even only one single species. But again, in most of the ponds, I was able to find some exuvia, except for the metal tub that I mentioned earlier. And in a few cases, you can also see that the amount of these exuvia was increasing throughout the project. So there was not much happening in May, but then starting in June, a lot of these individuals were emerging. I'm here now comparing the numbers of the larvae that I found and the exuviae in the different months for the different species here, and especially for Aeshna cyanea on the very left, but also partly for Sympetrum striolatum. We can also see the switch here. So for example, in Aeshna, in May and June, we had a really large number of larvae that we found. In May, no exuviae, but then in the further progress of the project, so in July and August, this number of exuviae went really high up, while the number of larvae that I found went quite down. So again, we can definitely see that there was the switch between the stages from larval to adult stage, basically, with exuviae just being kind of the proof that there was emergence. And then coming to the adults, to imagos, here in general, there was another pond where I didn't observe any imagos. That was the pond number 3, which was actually the barrel pond. This was a really, really small pond, and unfortunately, it was mostly in the shadow when I was visiting it. So it was not very surprising that I didn't see any imagos there, but at least I found larvae and exuviae. Here, I think it's quite clear that, again, the pond 7 was the one with the largest number of imagos that I observed, and also the largest diversity. And again, here we can see that there is this kind of seasonal change in the species that one could observe. In May and June, it was mostly the damselflies, so the Coenacronidae, which would be the Coenacron puella and alagma and ischnura, for example, and also Pyrosoma nymphula. While then later in the project, in, for example, July and August, I mostly found the Aeshna cyanea or, for example, Sympetrum striolatum. I can tell you these were like really, really very, very active, and that's something that is quite well reflected in the data. As mentioned earlier, I was also trying to figure out which of the factors may play a role for dragonflies. Unfortunately, I did not find much here. I also want to mention that obviously this is a really small sample size of just 10 ponds. There's probably a lot more variables that one could measure. So if I would do this again, I would definitely do more. For now, what I can show at least is that I looked into the species richness, so basically the number of different species per pond when it comes to vegetation and larvae. And I also looked into the diversity of the vegetation and larvae, which is calculated both from the number of different species, but also their actual abundance. And here we do not find any relationship. So from my data that I collected this year, I cannot say that vegetation or its species richness and diversity plays a role for the dragonflies and damselflies. Here, the only pond that kind of stood out a little bit was, again, pond number 7, which I can tell you is also the largest pond, but it was also definitely the one that was most diverse, that kind of occurred as the pond with also the highest larvae diversity and species richness. But as I said, apart from that, there was no relationship. I also looked into the relationship between the vegetation and exuviae. And interestingly, if the diversity of the vegetation was quite high, the number of found exuviae was low. So we find this like negative relationship between diversity of vegetation and diversity of exuviae. My idea is that actually this doesn't really has much to do with how important vegetation is for the emergence, because vegetation is important for the emergence. It's something that we know from other studies. But in this case, the more diverse the ponds were and the more covered they were in plants, the more difficult it was to actually find and collect exuviae. So this is rather my explanation for this negative relationship. Okay, and then summing this up, the results from this project, I could show that the diversity of larvae and exuviae was similar. but that the occurrence was staggered in time. So we had this switch between the between the different life stages. And I only observed very few imagos, so it's really difficult to kind of draw conclusions from that. When it comes to the species-specific phenology, yes, we did observe that, or I observed that in the project, especially when it comes, for example, to the common darton that had their peak emergence in August, while some of the other species emerged earlier. Several species co-occurred, despite the really large numbers of the Aeshna cyanea larvae. The reason why this is important to mention is because Aeshna cyanea larvae are really quite huge. They are very predatory. They're even very cannibalistic. And it was actually the assumption that if there is a large number of these larvae in the pond, that none of the other larvae can actually survive and that all the other species will just be completely outcompeted. But this was not the case. So I believe that if there is enough also vegetation and everything, and enough other diet, that these larvae can definitely also co-occur. When it comes to the biotic factors and the pond characteristics, I've still left it here as a question because I would need to spend more time with this. And as I forgot to mention it earlier, but I didn't analyze the water quality parameters. This is something I will still need to do. So at least in my project, this is definitely still the part that needs a bit more attention, and especially a larger sample size. But I can at least show you data from a different study that actually also looked into ponds, in this case, however, in Canada, and they were specifically looking into in how far three factors play a role for dragonflies and damselflies, being plants in green, land cover in brown, and the water quality in blue. And just very briefly summarized here, only for the dragonflies, we see a very similar pattern between the adults on the left and the larvae on the right. And in both cases, it's the plants and the water quality that play a major role, while the land cover plays a smaller role, let's say. We do, however, find a very different result if we look into the damselflies. Here, the land cover for the larvae doesn't seem to play a role. For the adults, it does play a role, but it's really a minor role, and it's really mostly plants and the water quality. What I just want you to take from that is that, yes, plants and vegetation do play a role, although this was not shown in my project itself. And also the water quality plays a role. It is secondary to the plants, but the role is still, I would say, significant. And that in this case, in this study here, it's also shown that this may be very specific for the different life stages, different groups. So basically the dragonflies and damselflies have different needs, let's say. And yeah, coming to an end, I wish to thank all of the participants and helpers of the project, specifically Tim, Johannes, Sarah, and Katharina for their help. I especially would have not been able to do this without Tim, who encouraged me from the beginning on to do this. Then I want to say thank you to the entire course from the Ken Art excursion that we did this year. It was really one of the best things that I did last year. Me enjoying my time there. And this is the photo that I really need to show, and I'm really grateful to Georg who took it because it was the moment that I saw one of these dragonflies emerging, and I was literally just like, people, stop whatever you're doing. You need to come and see this. And then we all spent kind of half an hour looking at this dragonfly emerging. So thanks, Georg, for that. This is also what happens during field work, just so you know. And I'm kind of famous for getting stuck in mud or something and people having to save me. It is what it is. And then finally, I just want to quickly mention the ID Logics identification app, which is a German app. It's developed by some colleagues in Hannover, and I'm currently also working with them. They have basically packages for different groups of species, including dragonflies, where you can answer questions about their appearance, and then the app will suggest the species that you've probably observed. And currently they have it for the imagos, and I am now working on the part for larvae and exuvias, so stay tuned. I really hope that we will be able to kind of publish this extra package sometime soon. And then finally, I want to end this talk with the citation. I'll just read it out loud. Although it is species themselves that typically have the greater impact on public consciousness when they are threatened with extinction, it is their habitats and the ecosystems and biotopes that contain those habitats that must constitute the primary targets of protection, because no species can persist for long without a suitable place in which to live. This is from Corbett from 1999, and I think that this is something we can all hold to also today. And with that, I would really like to thank you all for your attention. Many thanks again also to Lotta Domscheit, who helped with this project as well, and to Tim. Feel free to follow me on social media. Feel free to contact me anytime. And I'm really happy to receive your questions.
Maja, that was amazing. Thank you so much for this truly amazing talk. It was fun. It was informative. You were within the time limit. I mean, woo!
Awesome.
So off to questions now. Although I had also some questions of my own prepared, but you answered them all, of course, during your lecture.
Okay, good.
As expected. In registration form, there was actually one question about Zoenagrion ornatum species in Croatia, but for that I'm going to refer to our expert, Dr. Ana Štih-Koren. Ana, if you are here, you can you join in and maybe answer about this species here in Croatia?
Yeah, hello everyone. My name is Ana, and as Anja said, I'm working in Hyla. Maja, first, I want to thank you for this interesting and inspiring lecture, and well done on the project and your results, very interesting results. And I have to say that it's always nice to see so much passion that radiates from you. And your passion is certainly one of the reasons why so many people have joined us today. 48 people, Maja. And now we can get back to Coenagrion ornatum. Since the question was not specified, Anja, it was only
yeah, only about this species in Croatia. So it's ornate bluet in English.
Yeah, Coenagrion ornatum. Okay, then I can just briefly talk about its distribution and about its habitats. In Croatia, the species is present in all three biogeographical regions, in continental, in alpine, and in Mediterranean. However, I can say it's not widely distributed and it is missing from many parts of the country. For example, in Dalmatia, it occurs only on 3 localities, in the southernmost part of Croatia, in Konavle field. Then in the Alpine region, it's only present in Lika, but we don't have it in Gorski Kotar. Gorski Kotar is a mountainous and forested part of Croatia, so it is not present there. It is also missing from Istria. We only have few findings with the border with Slovenia. But in continental Croatia, the situation is different. It is much more widespread, except from the easternmost part of the country where it is also absent. So as a conclusion, I can say that it is very local species. Yes, thank you, Anja, for the map.
Yeah, I'm trying to share just from Biologer data.
but at the sites where it occurs, it is present in large numbers, relatively large numbers. And I'm speaking only about the localities where I personally found it and which I personally visited. So I never found only single individuals. There were always multiplies individuals present on the site, both males and females. And the habitats in Croatia, I found it only in continental Croatia, and there it inhabits typical habitats for Coenagrion ornatum, small, narrow streams with banks are not steep, mostly unshaded, streams that go through the meadows, slow flowing, and with a lot of emergent vegetation. So this is shortly about distribution and habitats about Coenagrion ornatum in Croatia.
I'm sure if there will be any other questions, maybe more detailed about the species, you can always contact us at Hyla or Anna or Maja. Our contacts are not difficult to find. I can also share them in chat. We have additional question from Rebecca. She's asking if Odonata species are nocturnal or active during dawn or dusk. She's asking because she's wondering about the effects of artificial light at night, and she's guessing that they are most active during the day because they are visual predators, but maybe there are some exceptions.
Yeah, thank you, Rebecca, for this question. Also, thank you for being here. I've actually already seen that Anne was commenting on this. I probably would have said something similar. I've also heard that there are some that are active, especially when it comes to the mating. Also, the emergence happens mostly, I think, at the dawn. But the mating and predation can definitely happen as well for certain species. So I think it is definitely worth looking into the effects of artificial light at night. Exactly. But yeah, I would not say way more than Arne already said. If anyone else has anything that they can contribute and add, I'd be grateful for that. But short answer is yes, there are some that are active at that times as well. Yeah.
Okay. Let's see. Okay, so Mario wants to know, do odonatas have any symbiotic or parasitic relationship with other species? That's an interesting one.
Yes, they do. That is the only thing that I can say that they do. I've seen. I also have pictures of mites covering kind of like half of the body of the damselflies, for example. So there is a certain mite. I'm not sure if it's very specialized on Odonata or not, but I've definitely seen this in nature as well. Not sure about the symbiotic relationships, but at least parasitic, I'm pretty sure there are examples of that in the nature. Yeah.
Anna, feel free if you have anything to add
Yeah.
for these questions. I'm really not the expert on Odonata, so I wouldn't know. Sorry. Anna is, I think, satisfied with your
Okay.
answer, so
Awesome.
everything's good.
Yeah.
A little bit before in the chat, we already answered the question about Simpecma pedisca, I will say.
Yeah.
Sorry
Yeah.
if I mispronounced it. Thank you, Angela, for that. She said that this is a species that also overwinters in Germany. She was referring to the species you mentioned in the lecture
Yeah.
as the only one that overwinters, but she knows about this one as well. I checked and Damian confirmed that Croatia doesn't have this species, but so this is resolved.
I'll just quickly jump in.
Yeah.
I just want to say thank you to Angela for being there. Angela is actually also one of our teachers from the course. So it's a little bit embarrassing that I said this wrong in the talk, but I'm really grateful to Angela for correcting me. And also, she's the author of this beautiful book that you can't see because I'm muting all of this, which is The Dragonflies of Germany, and it's one of my favorite books that I'm using quite a lot. So if any of the German participants here are ever interested in books, this is one that I can definitely recommend. And also, Angela is an absolutely amazing person, and I'm very, very grateful for all of her time and enthusiasm that I experienced in the course. So yeah, thank you so much for being here, Angela.
Awesome. I've also shared in the chat our mail that you can reach any of our experts via this mail. And if there are no other questions, I would like, yeah.
There is one from Jeka.
Sorry, I missed it.
No worries. First of all, thank you, Kira, for duckweed. I forgot about the word. I knew it's something with duck, but I wasn't sure. So thank you, Kira, for telling me that. So Jeka was asking, where are they during the night? I mean, into vegetation, right? I mean, mostly kind of just resting, hiding. Anna, is there anything else one can say about this? I would just say they're in the vegetation, in surrounding vegetation.
Yeah, in tall grasses, on shrubs, some species in trees. Yeah.
Yeah.
Wind protected sites, they are sheltered somewhere.
Cool, thank you. Now we're done, I think.
Okay. Of course, if any of you have any other questions, anything pops into your head in the later days, feel free to contact us. There are still some comments. I think that those are not questions, but interesting comments about parasites. We'll check that out. Thank you, Peter. Thank you, Angela. And thank you, Maja, again for this talk. It was amazing. Thank you, Anna, for joining in with additional information. Thank you all for coming. I hope everybody liked it. And feel free, again, to contact us. Drop us a follow on socials. Stay in touch. And have a nice evening. Thank you all, and bye. I'll end the meeting now. It's always awkward, but I have to do it. Bye.
Thank you, everyone. Bye-bye.